Outbreeding devices, pollen pistil interaction, artificial hybridization. Outbreeding devices. Now, why do plants outbreed? That's because continuous self-pollination can cause inbreeding depression. And inbreeding depression means loss of genetic variability in plants to adapt with changing environment conditions. So what are the different outbreeding devices adopted by the plants in order to prevent self-pollination? So the first way is pollen grain release and stigma receptivity is not synchronized, means they both are not at the same time. Anther and stigma are placed at different positions within flower. Then self-incompatibility. Self-incompatibility means pistils will reject the germination of self-pollen. And then comes production of unisexual flowers. Castor and maize, male and female flowers are present on the same plant. So they are monoecious. So they prevent autogamy but not geitonogamy. Since both the male and female flowers, though they both are unisexual, they are present on the same plant. That's why they cannot prevent geitonogamy, but they can prevent autogamy. Next example is papaya. For papaya, male and female flowers are present on different plant bodies. So in this case, they can prevent both autogamy and geitonogamy. So it will be a true cross-pollination. Here we can see the anther as well as the stigma. They are placed in different positions, like stigma is present over here and these are the anthers. So since they are placed in different positions, the chances of the pollen grains from this anther to fuse with the stigma of the same flower is rare. So this can promote cross-pollination, that means outbreeding. Now this is a maize plant. Here what we see in the tip of the shoot are male flowers and these are the female flowers. So the position of male and female flowers separately on the same plant, they can prevent autogamy, but in this case they cannot prevent geitonogamy. Now production of unisexual flowers in case of papaya, which is a dioecious plant. So the male flowers are present on one plant body, female flowers are present on another papaya plant. So from the male flowers, the pollen grains have to go for cross-pollination or xenogamy in order to facilitate fertilization. So in this case, there is true cross-pollination or outbreeding. Next is the topic pollen-pistil interaction. It means that all the events starting from the pollen deposition on stigma until the pollen tube reaches the ovule. So pistil has the ability to recognize the pollen. If it's a self-compatible species, then pistil will accept the pollen and allow the pollen tube formation. But if it's a self-incompatible species, the pistil will reject the self-pollen. What will happen to the pollen grain if it's accepted by stigma to germinate? So if the pollen grain is accepted by stigma, it will germinate on the stigma and it will produce a pollen tube. The pollen tube carrying two male gametes will pass through the style and they will reach the ovule. So there is filiform apparatus present on the synergids here, present near the egg, and filiform apparatus will be guiding this pollen tube to reach the egg. And then here double fertilization will happen. Like in this pollen tube, there are two male gametes present. One male gamete will fuse with the egg and that is called syngamy, leading to the formation of zygote. The second male gamete will enter into and fuse with the two polar nuclei that's present in the central cell, and that will result in triploid endosperm formation. So since there are two types of fertilization happening here, it's called double fertilization. So one fertilization is called syngamy here, when the male gamete fuses with the egg. Second fertilization, in which the male gamete is fusing with the central cell, that is called triple fusion. Why? Because that fertilization results in triploid endosperm. That's why. The next is artificial hybridization. Artificial hybridization means crossing different varieties of plants to get a desired hybrid. For example, a cross between red rose and a yellow rose in order to get a hybrid rose. So what are the steps in artificial hybridization? The first step is emasculation. So if the flower which is chosen as a female flower is bisexual, then it has to be emasculated, which means the anthers have to be removed. See, here in this case, the anthers are removed with the help of forceps. And this emasculation is done at the bud stage. Now, if the female flowers are unisexual, then there is no need of emasculation, as anthers are already absent in that. The next step is bagging. After emasculation, the female flower buds are covered. They are covered with butter paper in order to prevent contamination of its stigma with unwanted pollen grain. So this process is called bagging. And the third step is called rebagging. Once the stigma becomes receptive, the desired pollens are dusted on stigma and then they are rebagged in order to prevent contamination from unwanted pollen. Fruits are allowed to form, then their seeds can be sowed to get the hybrid plant. So after the rebagging, the fruits will be formed within the bag, and then from the fruits we will get the seeds, and these seeds are hybrid seeds. So that's how artificial hybridization is done.